So this is a presentation from me and my colleagues working on the calibration validation project of the PRISMA. So we have the potential, we have the opportunity to work with real PRISMA data. Initially, I will give you an overview of what is non-photosynthetic vegetation at large, and then specifically talk of PRISMA data potential. So starting with the definition, NPV is vegetation that can no more convert solar energy into chemical one. So it's composed by standing, dead or dying vegetation, plant litter, and crop residue. And so looking at the literature, the importance of NPV is connected there because it plays a role in the cycling of carbon, nutrients, and water, and is important in soil conservation since it increases the carbon attraction and water holding capacity in soil while reducing the runoff and erosion. And in the literature, there are too many study branches about NPV. One pillar is ecosystem functionalities. In this pillar, it is a real variable. It is biomass gram per square meter. And the most common objective is cost per estimate and grazing effects. The other pillar is the one I'm talking about, is sustainable agriculture. In this case, the variable is most like cover fraction or class of cover fraction. And the most common objective is to monitor conservative practices, in particular tillage practices. And this is also considering the eligibility for subsidies asked from farmers. So the EO data in this case and in many other applications has been used because they are more rapid, accurate, unbiased, and more economic as well. And most of the literature deals with optical remote sensing and on distinguishing three main categories, which are non-photosynthetic vegetation, green vegetation, and bare soil. The basic concept is this part of the spectrum is dominated by pigments, and this part of the spectrum contains information on non-pigment plant constituents like proteins, lignin, cellulose, and starch, and so something we are interested in. Multispectral data have been exploited for the detection of NPV, and several spectral indices have been designed to that purpose, and the most common is the normalized different tillage index, which has been designed for the lens emissions and so in time. And this detection is quite good, but the characterization of the fraction of crop residues is not so good because there is a lack of for proper spectral bands of the proper resolution in this shortwave infrared for an accurate characterization of the variable. Really interesting. So going to hyperspectral data is the best choice because in the shortwave we require a spectral resolution which can help us to resolve the NPV absorption features, which are quite narrow and subtle sometimes, and also because they overlap with other variables, so we have to disentangle the effect of, for instance, water vapor. So there are again some literature, there are again some spectral indices, and most of them exploit the lignin cellulose absorption peak at 2.1 micrometers and the lignin one at 2.3 micrometers. So the most common ones are cellulose absorption index and the lignin cellulose absorption index. But as has been told also for the lecturers, the time is changing, the perspective is changing. Now we have missions and a lot of missions. So such indexes were designed especially for ASTER and IP data. Now we are moving to a new era. So moving from spectral indices to real spectroscopy to be more quantitative, and to make that, we have to take account of all factors affecting the NPV signal in a particular water content. And this could be improved also by the modeling of biochemical properties, while it includes also carbon-based constituents, like it has been done by the ProSpect probe. So going to PRISMA first results, as members of the PRISMA project, we have the opportunity to work on two test sites in which we collect field data for the validation of PRISMA. One is in Slovenia, in South Korea, and the other one is Yolanda, but this has been also part of the iPresence campaign this year. Both two farms are very large. The first one is around 1,000 hectares, the second one around 4,000 hectares, and they have a lot of different crop types. So for this is interesting. We made our first experiment just to map and to be against other classes, and basically on the band presence, the band of cellulose lignin. And we made it just with a quadratic approximation, in which if the coefficient A of the approximation is positive, so there is the band, there is the absorption peak, and so it is classified as NPV. Otherwise, is vegetation or soil. We use the crop map of the farm and also our measurements on the ground as a ground truth. And so, for instance, we made this map, we produce this map, which is on the parcel level. So we use the average spectra of each field. And we see that this, even if rough, it's a good approximation, because they are looking at durum wheat, which had been harvested at the time of overpass. Everything was correct, and both dots, which are with a negative A coefficient, correspond to fields where were already tilled, and so it is correct. It's soil, as it's not NPV. And so we decided, starting from that, to go on this peak and try to measure the band depth to have more information in a more quantitative point of view. So at first we try with this approximation, which is really an optimization with the Gaussian function, which is also exponential to take account also asymmetry and saturation. And we have good results. First results that told us that two things: that the band is not sensitive to albedo and bidirectional effects. In fact, if you look at both box plots, are the same parcel, which has been acquired on two subsequent dates with two opposite looking angle from the PRISMA, and so we have quite a good consistency in between the 2 dates, which is sensitive to the band depth, is more related to the species or varieties. Those are different durum wheat varieties, and this is barley and this is soft wheat. And this band depth works really good with field data when it comes to estimate the fractional crop residues. So we are encouraged to go on and understanding the potential of this band, but not of this band alone. We decided also to, since on the ground there are different conditions, this is not only crop residues and other stuff, but there are also crop residues with little vegetation, which are typical after a minimum tillage. There is plant emergence, and so there is a mix in between MPV and between green vegetation, as well as there is standing dead or dying vegetation in one image. So this is Yolanda, this is Hypersense campaign last June. And if you look at, so we consider three different absorption peaks. One is the lignin cellulose one is related to pigments, to chlorophyll, and the other one to water content. And if you look at this scatter plot in between lignin cellulose absorption peak and the pigments one, you can see that, for instance, there is a difference between crop residues and crop residue related vegetation, because there is some pigments in this, and there is a difference also related to pigments in between crop residues and standing dead vegetation, because standing dead vegetation has still some pigments inside. And bare soil seems to have no peaks in the two pictures, while green vegetation has, of course, pigments and very and quite narrow MPV absorption peak. And so crop provides with little vegetation is something in the middle, and this could be distinguished also on the RGB composition of the three of the different pictures, and this is allowing us to apply a rule-based mapping to have this different five categories on the ground. And so after that, we can work only on those two, which are the ones we are interested in, to quantify the fraction as a variable of MPV. So our experience with PRISMA data prove that the satellite is in the center is suitable for mapping MPV, and using lignin cellulose absorption peak, but not that alone. Also the other ones which are affecting the crop residue reflectance. We will keep on with modeling for different species to understand the differences in between species and sites and in time, of course, since we have this quite good time series of PRISMA images. We will look better to the atmospheric corrections, to sensitivity of the method to atmospheric correction, to include also other features, especially regarding soil. And we will go on and test the model with other remote sensors as the ones of the Hypersense campaign, and of course in the perspective of the cruise emissions, as has been said before. Thank you for your attention.